Hello, class 12. We will be starting with the principles of inheritance and variation and continuing a genetics path, the chromosomal theory of inheritance and the sex determination today in this chromosomal theory of inheritance. Sutton and Boveri proved the chromosomal theory of inheritance and they proposed it actually, and the Sutton is credited with the first proposing the chromosomal theory of inheritance. Now they worked on the chromosomes, the meiosis part, that chromosomes are in the pair. The homologous chromosome separates during meiosis so that alleles are not segregated, means they separate, and that is why there is a variation. And they also prove the process of meiosis. Meiotic products have one of the each homologous chromosome but not the both. Fertilization restores the pair of chromosomes as they meet again, and genes are located on the chromosomes that we know. This is a example showing over here the chromosomal theory of inheritance of the yellow and the round seeds with the green and the wrinkled seeds. During the meiosis, the gamete formation and fertilization, the F1 is proving the principle of inheritant assortment and also the law of segregation. Also, the ratio is coming 9 is to 3 is to 3 is to 1. Fertilization is resulting, leading to the phenotypic character. Independent assortment of the chromosome. This is the figure of the NCERT where the meiosis and the germ cell formation in the cell with the 4 chromosome, not how the variation can be. Now this variation where the two possibilities of the different types of the germ cells can be. This has been beautifully shown with the help of the different colors used in the chromosomes. A diploid chromosome over here where it has got form of the 4 haploid cells with the differentiation in the color pattern. This is a comparison in the behavior of the chromosomes and the genes. Go through it very carefully and suggest which one is belonging to chromosome and which is a gene. I would like to give you a hint over here that genes are present on the chromosomes. Now, Sutton and Boveri, who have gone through this chromosomal theory of inheritance, Thomas Morgan has worked on Drosophila melanogaster. And what was the reason behind he chose Drosophila melanogaster? The first important reason was that it could be grown on the simple synthetic media in the lab. Secondly, they complete their life cycle in about 2 weeks. Third, a large number of progeny are being formed. Next, there is a clear differentiation of the male and female Drosophila, easily distinguishable as the male Drosophila is small and female is bigger. That's the reason, and also has many types of heredity variation can be seen with the low power microscope. So this forms a very important question that why T.H. Morgan have chosen Drosophila melanogaster. What is genetic linkage? In this slide, it is shown the genes which are present on the chromosome and the genome size of the humans. Over here, the linkage and the recombination, when the genes are present on the chromosomes over here and the distance between the two over here is low, so there occurs the linkage. But when this occurs between the two nearby chromosomes, then recombination occurs, which leads to crossing over, which leads to more new variation. In this diagram, it's beautifully seen by the homologous chromosome pair in the prophase 1 and in the crossing over, how the crossing over is occurring. For this understanding, you should go through the chapter of cell division in cell cycle of class 11. This is a test cross of the Drosophila over here, the F1 hybrid gray and the normal wings, and with the meiosis and the double mutant black with the vestigial wing. It is seen, and the recombinant frequency. I know that this is not easily understood to you because it is not easy to see, because over here we are just studying what the results we are getting. So you should over here know what is test cross, how the gametes are formed. This thing is important to you all, and what is recombination. This is again the diagram same and explaining the recombination. This is exact diagram of the NCRT, which is representing the linkage and the recombination results of the two dihybrid crosses conducted by Morgan of the cross A between gene Y and W, and cross B showing the crossing between gene W and M. Now here the dominant wild type alleles are represented with the plus sign, superscript, and next with the minus sign. Now just go through over there, and the recombination 1.3% and recombination 37.2%. In the both the crosses, the results are different. The results are different because of the linkage and the recombination. A long question comes to describe this, that what is linkage, what is recombination. This is exactly the figure of the NCRT. So go through the how the gametes are formed and how the linkage and recombination. I know the problem will occur with the recombination and linkage, but you should know what the results of the yellow and the white, and white in miniature is coming. Wild type is the pure type. The sex determination. Hanking, who discovered the X body or the X chromosomes. This is a diagram of the honeybee. Honeybee, a great saying is there for the honeybee, that the male honeybees have the grandfather, grandmother, but they don't have the fathers. Why it is so? Because in honeybees, the male are haploid and the females are diploid. Now, the females are formed because of fertilization, and haploid male bees are formed without fertilization. Okay, so what will happen? Now, the haploid male gamete will have the mother, as they are forming from the diploid female bee. Yes, it is true, without the interference of the father, but that female bee will be having the grandfather and the grandmother. Just go through the saying, go make the Punnett square of it. Male heterogamity and female heterogamity. Hetero means different. So over here, the male component which will have the chromosomes in the heterogamity will be called as the male heterogamity, and those female who will have the different heterogamity, that will be called as the female heterogamity. So the male who is producing two different types of the sperms, while female produces only one type, that is male, and the female which is producing two different types of gametes. Example is grasshopper, bugs, XY. In humans is also example of the male heterogamity. ZW type, one of the heterogamity, birds, reptiles. It is a very important question in the academics. MCQ can also come from this topic. Heterogamety means different sex gametes, includes the human males, including XY chromosomes is there, grasshoppers are there. Female heterogamety includes female of some species like birds are there, fishes are there, insects are there. Females of butterfly, moths, that is ZO, and females of fish, reptiles, ZW. This is a sex determination in human beings that you also must have studied in class 10. Go through it, because you should know how to make the Punnett square of it, and what are the autosomes, what are the germ cells and the sex cells. Independent assortment of the chromosome. This is the figure of the NCRT, where the meiosis and the germ cell formation in the cell with the four chromosome, not how the variation can be. Now this